Hello and welcome to this lecture on fat digestion. Real quickly, we'll go through a basic overview of fat digestion. Similar to pretty much everything else, we will see some digestion beginning in the mouth, with particularly mechanical digestion to begin to break things down through chewing and moving things around the mouth with the tongue. We do have some enzymes, lingual lipase, in the saliva that will begin to do some breakdown of fat molecules. But primarily when we move into the stomach, that's where we see really the beginning of the bulk of fat digestion. So we do have some gastric lipases, again, enzymes that will work on fats in the stomach. And again, we have mechanical digestion in the stomach with the churning. Those kind of smooth grooved muscles in the stomach will turn things over and turn, and thus help to break down the fat molecules and move the products of digestion, the intermediate products of digestion down into the small intestine. And then in the small intestine, this is where we see the secondary major source of fat digestion. So this is also where we see bile being made by the liver, stored in the gallbladder, and then secreted from the gallbladder into the small intestine. And remember, bile is basically a conglomeration of phospholipids, different types of phospholipids that help to emulsify small fat molecules, basically cholesterol, sterols and triglycerides, so that we can package them in such a way that they can transport through the blood and be delivered to different body tissues. So let's kind of not necessarily go through that again, but just kind of look at a different schematic and focus on the small intestine. So again, we do see some amount of chemical digestion from the mouth and the stomach, but then really it's the action of the bile produced by the liver, stored in the gallbladder, secreted from the gallbladder into the pancreas. And so, and then the work of pancreatic lipases. So again, those three accessory organs playing a really big deal in the eventual digestion of fat in the small intestine. So if you look at the diagram in the bottom, this is inside the small intestine, basically. And so you'll see kind of the schematic showing this light blue up top, the digested lipids and the bile acids. Again, these would be phospholipids surrounding those digested lipids. So again, working with a hydrophobic aspect to the bile and a hydrophilic aspect to the bile, such that it can both interact with fats and water at the same time. So as the fat is digested, it will be absorbed, the components of it will be absorbed, just like we saw with glucose, galactose, and fructose will be absorbed into the mucosal cell or the enterocyte. And really what gets absorbed are fatty triglycerides and cholesterol esters, basically. And so there's this term back up here, the micelle, which is basically what we have, basically the term given to the process of fat digestion, it is the molecule basically of bile acids acting on fats as they're being digested in the lumen of the small intestine. And then we have this other word, chylomicron, which is the once inside the enterocyte. Again, the individual components of fat digestion, primarily cholesterol esters and triglycerides, will get packaged together into a chylomicron, and the chylomicron eventually leaves the enterocyte into the lymph, and the lymph will ultimately meet up with the blood supply, and thus the chylomicron will deliver cholesterols and triglycerides to various body tissues. Not a ton of fat would get excreted eliminated in the feces unless there was a huge excess coming in dietarily. Particularly, remember, we do manufacture our own cholesterol. So when there's a huge excess of cholesterol, a healthy way to get rid of it is with whole food sources of fiber, and the fiber would bind the cholesterol and help eliminate it through the stool. And this is, again, just a really similar schematic of the other 2 pages, but again, kind of showing the micelle basically being this conglomeration of both the bile acids and the components of fat digestion, and then those individual components, basically free fatty acids and triglycerides or diglycerides or monoglycerides, would then enter into the entire. Oops, I didn't mean to click onto the next page, but if I go back, it erases. So I'm just going to keep going forward because this is, again, just showing you through a different schematic, the same exact thing. But here we actually see the micelle. It kind of shows the two different aspects of the micelle after the full emulsion process with the bile salts. So the cholesterols and the monoglycerides and fatty acids individually enter into the enterocyte. And then they will pass through both the endoplasmic reticulum, the smooth ER, and then the Golgi apparatus to be packaged once again, or not packaged once again, but packaged for the first time really into this chylomicron. And again, there's kind of such a process here, both in the emulsification or breakdown of the fat droplets and in the packaging before ultimately entering lymph, which would then go into the blood system, because we're talking about fats in a water environment, so they have to necessarily be packaged with basically types of phospholipids, again, to allow them to be both hydrophilic and hydrophobic. And sorry this is so blurry, but just again showing the structure of this is the chylomicron, what would be transported through the lymphatic system, delivering triglycerides and cholesterols to body tissue. And you'll notice the outer membrane is phospholipid, so that the chylomicron can pass eventually through the blood supply. And then this is just kind of showing, without the full imagery, kind of showing dietary fats coming into the body, getting packaged at the small intestine into chylomicrons. And again, the chylomicron is effectively carrying dietary triglycerides and fat cholesterols to muscles or fat cells. Especially in the fat cells, we would see the fat molecules, or in adipose tissue, we would see the fat molecules being stored. We might also see that the triglycerides or cholesterol might go to heart or muscles for oxidation to provide energy. But what we might also see, if there is a huge excess, this is where we sort of run some risk in plaque development in the arteries. So then you'll also notice, if there is excess, if we have this molecule, high-density lipoprotein, high-density lipoprotein does something that is kind of the reverse of, it's kind of clearing cholesterol out of the body. So if we do have excess cholesterol, high-density lipoprotein, if well and functioning well, will deliver excess cholesterol to the liver. And here the liver has the capacity to basically say, okay, we have too much cholesterol, so let me get rid of cholesterol, basically sending it back into the large intestine to be packaged into stool and eliminated. And then I want to look, if we just look through this way, straight down through what the liver can do, because we looked at what happens if we are taking in dietary fats, fats coming in through our food supply. If we start here at the site of the liver, remember, as I said in the previous slide and in the lecture on introducing fats, our body has the capacity to make cholesterol. So taking cholesterol in through our food is not necessary to do so. Although cholesterol is an essential molecule to human existence, we don't need to eat it in our food supply because the body makes it. So when we make cholesterol, the liver will send out what are called very low density lipoproteins and low density lipoproteins, again, to distribute cholesterol or perhaps triglycerides to areas of the body when they need them. So particularly, say, heart and muscle for oxidative work, which is making energy. So we already have this sort of natural system of making cholesterols and delivering them to the body as needed. And low-density lipoproteins are typically associated with excess cholesterol. So if it were the case that we had excess coming in through the food supply or some sort of issue where the body was making excess cholesterol, which is not very common. But anyhow, if we wound up with an excess of cholesterol, then low-density lipoprotein oftentimes is would be carrying that excess cholesterol. And again, that's where we sort of run the risk with having so much excess and not even being able to utilize it for oxidation because we're not burning enough energy, we're not utilizing enough energy through our daily expenditure. We may see the deposition of excess cholesterol in the arterial walls. And then I think this is the last slide for this presentation, but just looking at the lipoproteins. So again, you notice that these are both hydrophilic and hydrophobic. Hydrophilic on the outside with the phospholipids making this membrane, and the phosphate head facing out towards what would be the watery blood out here. And then the hydrophobic, or the lipid, the fatty acid tails going inwards, and inside there is where we have packaged basically cholesterol, free fatty acids, and triglycerides. So again, inherently not a bad thing. It's just it again comes back to just balance. I mean, the body naturally packages these lipoprotein molecules to deliver triglycerides, cholesterol, free fatty acid around the body as needed, and or when they're in excess, bring them to the liver to be packaged out of the body. So really, it's just a matter of balance, as we see in so many aspects of this course.